Hello and welcome back. This is Dr. Swope taking over for Mrs. Fenton. If you've been with us before, or if you're new, from Germantown, Maryland. I teach at Northwest High School, and if you've been with us before, I promise not to say it. So here we go. Let's dig in. To where Mrs. Fenton talked about on Monday. She went into what the AP psychology exam for 2020 was going to look like and shared some information. And I thought if maybe you missed that, or if it was a couple days ago, a little refresher wouldn't hurt. So, as you probably know, for the AP psychology exam for 2020, we're only going to cover the first 7 units just because of where the virus took over and many of us had to leave school. Learning was a little bit hiccuped or had an interruption. But you'll remember the first couple days of this review, Mrs. Fenton talked about clinical psychology, which was, of course, abnormal psychology and treatments and therapy. Now, on the last day of this video review series, Mrs. Fenton and I on May 1st will talk together. We're going to appear together and go over some of the most important units and concepts in social psychology. So again, the what is the first 7 units. The when is May 19th. And more specifically, you've probably heard about that, but if you're watching this, it might be worth you to jot down your time or take a screenshot, or of course, you know this is recorded and you come back to it when your particular time zone, when your exam will start and when you'll log in. Okay, so that's the when. Let's talk a little bit more about the how and more what. So if you're new to us, let me go over it quickly, or even again, if you've seen this before. There's going to be 2 questions. You're going to have 25 minutes once you open the app, once you log in, to do and upload question 1. Okay, so that's 25 minutes. And then question 2 is 15 minutes. And you'll notice the weight is a little bit proportional. What I want to urge people to do, and I've heard some from my students and other teachers I've talked to say, oh, well, we don't have to study, it's open note. But I really want you to take a look at this 25 minutes mark right up here. And if you have, let's say, 7 terms that you're going to need to define, apply, and add a synonym to, that's going to be 3 minutes per term. Maybe a little bit more, but you get the idea. On average, 3 minutes per term. You want to start this with a pretty good bank of familiarity in your own mind. You really want to make sure you keep studying, because sometimes there's a misnomer with open notes. Like, well, I can just look it up. I can use my notes. But time is really going to be a factor here, especially if you're flipping back and forth through different resources. So again, I urge you to keep studying because time's going to be a factor. When we look at question 1 and they call it concept application, that might be what maybe you're used to in studying before. I don't want to say it's just vocab, but it's going to be a little bit more content heavy as compared to the research methods, which might be a little bit more process or skill oriented, where we're looking at research methods, independent, dependent variable. Can you find confounding variables? Can you compare and contrast which research methods should be used or which ones are being used? So that's just a quick review of what Mrs. Fenton talked to you guys about on Monday. Let's move forward a little bit into what we're going to do today. Today we're pretty packed. You can see we have 5 bullet points, and what we're going to do is shift from the last 4 days were about biopsychology, brain and neurons and hormones and scanning technologies. And so today we're going to shift into what most people consider sensation and perception. And we're going to be doing that for the next couple of days. I'll be with you for today and the next 3 days next week. So for these 5 bullet points, describe general principles of organizing and integrating sensations to promote. That's a lot of words to discuss. What we're going to do is define sensation and how it turns into perception, because sensations and perception, while they overlap, are 2 very different things. And then in the second bullet point, we're looking at this word transduction. And within that, we have 4 or 5 very important concepts: absolute threshold, difference threshold, signal detection, and sensory adaptation. And we're going to spend probably a slide each on all of those. Major historical figures, what I did is I wove them into other slides. This isn't necessarily a class on the history of psychology, but we can't ignore how our knowledge got to where it could be. And remember, we are where we are because we stand on the shoulders of giants. Vision process. As Mrs. Fenton went over parts of the brain, we're going to go over parts of the eye, what they do, where they are, those kind of things. And then the last one, explain common sensory conditions. Okay. So Let's pick up from yesterday where Mrs. Fenton left you off, and it's a 45-year-old man reports to his doctor. Remember, this is the FRQ from yesterday, and I hope you took a little bit of time to actually jot this down, to give it a try instead of just kind of mentally saying, I got it, I got it. Remember, the more you practice, the better you're going to do. And at this point, maybe you want to start doing some of these FRQs timed. And get yourself used to that crunch of 25 minutes. Define, apply, add a synonym, and see if you can then kind of mock upload it. Because as we know from emotion, when the pressure's on and when you start to get stressed, maybe practice is going to help you out. The more you practice, the less you have to think. Okay, well, back to the FRQ. A 40-year-old man reports to his doctor that he's having trouble staying alert during the day, even though he gets about 9 hours of sleep on most nights. He has a high stress job and lets his doctor know that he smokes cigarettes and uses alcohol. During the day, he frequently finds himself falling asleep at work, and he admits he smokes more to try to stay awake. The doctor decides to conduct a sleep study to determine the reason for his problem. So part A, and let me stop here. It's really important to read the questions and not just blindly go in to define, apply, because the application is going to be dependent on what the question asks you. So explain how each of the following is related to the man's behaviors or symptoms. You got stimulant, you got melatonin, and then part B, explain how each of the following is related to the sleep study ordered by the doctor, EEG and sleep apnea. Okay, let's dig in. Well, if you've been with me before, I broke it down into apply, define, and synonym. This one is a little bit more streamlined, and you have some flexibility as to how you answer it. Of course, even within the SOTAS model, you can apply the SOTAS model in different ways. So stimulant. Because the man has trouble staying awake at work, he uses nicotine, a stimulant, to his body to increase alertness. When he smokes, the nicotine increases his heart rate and his overall level of alertness. Okay, even though this looks relatively, for lack of a better word, light, there's the definition, there's the application, and there's the synonym, kind of demonstrating no more. So nicotine, stimulant, there's the definition. And because we're linking it back to a man staying awake at work, that links it back or applies to the question, then that's definition, that's application. And notice when he smokes, the nicotine increases his heart rate and his overall level of alertness. So that not only defines and applies, but goes a little bit deeper. Now, the note here is that we must go specifically identify nicotine as a stimulant and as a specific physiological response, alertness, heart rate, those kind of things. It might not be enough just to say nicotine is a drug or nicotine is a stimulant, because that might be kind of just restating the question. Do not score. Now, a quick word on what scores and what does not score. Until the readers get the rubric and we know what the questions are, it's going to be tough to say what exactly scores. But if you say something that is directly contradictory to the information at hand and kind of contradicts yourself, that will not score. So if you say alcohol is a stimulant, or if you were to say nicotine is a depressant, that would not score. Melatonin. Again, let's read how we get the definition, the application, and the synonym all kind of in one smooth sentence. The man has been having trouble staying up during the day due to an imbalance of the hormone melatonin produced by his pineal gland. The man is having trouble managing his sleep-wake cycle because of an imbalance in the hormone melatonin, which is produced by the pineal gland and regulates sleep. These answers, these sentences are going to be enough. Okay, it identifies that it's a hormone. It identifies that it is involved in sleep-wake cycle, and identifies the biological source of that, and that's the pineal gland. Okay, let's go on to the next two. Now, this one we decided to do a little bit differently. Instead of me telling you what scores, why don't you take a quick look at this and you tell me, or I'll read it, but you're going to try to figure out which one of these would score. The doctor ordered an overnight sleep study to measure his sleep. The doctor had the man evaluated with an EEG that involves watching the man sleep. This involves scanning his body and his peripheral nervous system to determine which processes are or are not happening in the man's body as he passes through different stages of sleep. Okay, that is a response to the particular section of the FRQ. The other one, the doctor ordered an overnight sleep study to diagnose the problem. The doctor had the man evaluated with an EEG that involves a series of electrodes that are fixed to the scalp to measure his brain activity during the sleep. The doctor would be able to see the pattern of brain waves throughout the night and look for unusual patterns that would lead to a diagnosis of sleep-wake disorder. And I think we're going to see that it's the second one that scores. Okay. The doctor had the man evaluated that. If you look here, an EEG, I'm sorry, jump. So the second one scores, and let's talk about why the first one doesn't. Look at this second sentence. The doctor had the man evaluated with an EEG that involves watching the man sleep. It scans his body, and that's not enough. It doesn't help define what an EEG is, what it does, how it does it. It's based on electrodes on the scalp that measures the electrical activity of the surface of the brain. So the first one would not score, the second one would. Okay, for sleep apnea, as a result of the sleep study ordered by the doctor, the man was given a diagnosis of sleep apnea. The doctor explains that based on the sleep study, it is evident the man has frequent interruptions in his breathing during sleep, resulting in poor sleep quality and daytime sleepiness. Okay, compare that response with the second one. Sleep apnea is a type of parasomnia, which is a category of sleep disorders. When people descend into deeper sleep, their motor neurons are inhibited. Additionally, some involuntary muscles, such as those involved with keeping the airway open, are inhibited. Thus, a person's throats will collapse upon itself, which prevents air from getting to and from the lungs. Okay, which one scores? Can you predict? It's going to be the top one. The second one, while offering a pretty deep evaluation of what sleep apnea is, and it definitely offers a synonym, doesn't apply it. It doesn't apply to how the doctor's treatment of the man. So therein lies the issue. You want to be able to clearly define and clearly apply. Okay, well, that's enough from yesterday's FRQ. And again, I urge you with today's FRQ, take a couple minutes, write out the responses and time yourself. If you're showing that you're not going to be able to hit that 25 mark, well, you know what the answer is, and that's going to be to practice more. There's plenty of FRQs available, and I'm sure you can ask your teacher for more practice. Okay, principles of organizing and integrating sensation. Well, first, let's define sensation and perception. So to make it easy, I'm going to simply say that sensation is just gathering raw data. It's just gathering information. That's what your eyes do. Perception is processing the data into information. So if we could say sensation's gathering raw materials, perception is creating the finished product of a perception, which leads to a thought. Okay. The big question in these next couple slides in this first section is how. How does the physical brain and the mental mind do the process of reacting to vibrating air or frequencies of light? If you're listening to me or watching this presentation on YouTube, really all you're hearing is vibrating air molecules. Actually, you're not even hearing it. You're sensing vibrating air molecules, and you're not seeing the words. You're seeing light, but you're perceiving words. There's going to be the difference. See, understanding what goes on in the ear or the eye is relatively easy to understand right here. But what goes on in the brain and then the mind, that's the tough part of sensation. And that brings us to something called the binding problem. And it's really a question that no matter how much we know of neurology, psychology, and psychiatry, we don't know. We don't know how does sound and light from this video get merged into your mind in one conscious event. Up until I read that to you, this was relatively seamless. Now you might be thinking, oh yeah, how does words or sound create meaning? And that's transduction. And transduction is related to the binding problem. Transduction is the process of taking these raw material and turning it into perceptual information. The next one is the law of Prägnanz. The mind, or we, and depending on, I don't know if we can consciously do it, but say the mind unconsciously or we consciously, we prefer and automatically perceive things in the simplest way. Okay, so here's my example. I'm going to try to present 4 shapes. Okay, and I bet your mind or you unconsciously and automatically are going to perceive them in a way that makes sense. And I don't think you have a choice. Do you see four unrelated shapes? No, I don't think you do. I think the law of pragnance is a kind of a label of how our mind automatically groups things in ways that make sense to us, and we can't unsee it. My challenge to you is to not see a smiley face. Try to see an arc, a circle, and two dots. And it's very difficult for us to do. Okay. Bottom up processing is what I just asked you to do. It's noticing the individual aspects and assembling them into a coherent whole. So, for example, when you're listening to your favorite song, bottom up processing would be noticing, sensing, perceiving, and thinking about the notes, and then understanding it's a song. Okay. Or similarly, if you wanted to appreciate the beauty of nature, bottom up processing, would you go walk in the woods and you'd notice each tree, each flower, each leaf, and then assemble that into a whole forest? Top down processing is how most of us perceive and organize things most of the time. That doesn't mean that in some situations your brain, your mind doesn't do a bottom up concept. But for top down processing, it's we see the whole picture and then with effort, we notice individual aspects of that perception. When you hear your favorite song, you notice the totality of the song. You notice the whole thing. Or when you look at a forest, you look at the whole forest. You don't just say, oh, look at the bark on that tree and that specific square inch of that tree. And that's just not how we do it. So we mostly do top down. And in rare cases, we can do bottom up processing. Okay, gestalt, and that's how you pronounce it. There's a hidden H in the word. It's gestalt, and it's a type, it's, I guess it's rules or procedures or ways that our brain or mind automatically and unconsciously organizes incoming afferent sensory information. Let me quick take a break. Afferent is a word for signals that come to the mind. So we want to kind of in your mind link the word afferent and sensory. And what I tell my students is afferent signals arrive at the brain, efferent signals exit the brain to the peripheral nervous system. They're going to tell your motor neurons to flex. But going back to the idea of gestalt, and it's the idea that our brain assumes things or our mind builds a whole. So what am I talking about? I kind of talked around. So let's give you an example. Let's go back up here to bottom up processing, and I'm going to give you about 5 seconds to read that. Okay, if you were reading along, I don't think there's any words in there that you didn't know before, but did you count the letters? Did you notice how many vowels? I don't think you did. I don't think you noticed the letters. I don't even think you noticed the words. What you did was you got the meaning. You just, your mind assembled random light sources coming off of your screen into a coherent meaning in your brain. And so the quote with Gestalt is the whole is more than the sum of its parts. The whole is more than the sum of its parts. And for instance, if you look at Dr. Frankenstein's monster, he assembled, a foot and a leg and an arm and a torso and a head and got a brain, and it got struck by lightning, and bam, it could walk around and it could groan and moan, and it was a monster, and we know that. But was it human? Similar to the idea that what is a human is more than just all of our body parts. What is meaning is more than words and letters. So that's the idea of Gestalt. And that's how we perceive the world. We use kind of Gestalt rules or cues to make sense. Okay. Some of the Gestalt rules are here on this screen. Okay, but again, I want you to get the idea. Gestalt is ways that our brain mind automatically and unconsciously organizing information. So let's look at the Gestalt principle of proximity. When you look at this box, I would imagine your brain automatically grouped 4 and 2. It's very difficult for you to group a top row of dots and a bottom row of dots. And that is going to show that this is automatic. Okay. Your brain and your mind make sense of the sensation of light coming off your monitor. Similarity, we're going to group filled in, you're automatically group 3 hollow circles and 3 filled in circles. You're just going to automatically group them, and you're going to see 2 groups. I don't think you'd see 3 groups of 2, 1, and then the middle group of 2, and the right group of 2. Let's look at continuity. You perceive an arrow, even though you sense different shaded shapes, differently shaded. So you get a black shape, and then you're going to have a white shape, black shape, gray shape. So you don't sense an arrow. but your Gestalt rules are going to make you perceive something. Similar to the idea of this word up here, of course, you're going to perceive it as psychology. But what are you sensing? You're sensing a lot of broken letters. My students know that I have horrible handwriting. I don't think I've ever closed an O or a P in my life. But even when I write on the board, my students kind of get what I'm saying because of context or because of the idea of these Gestalt cues. You just kind of top down, well, we're in a psychology class. Of course, that word means psychology. Common fate. Perhaps you've been driving and two cars speed by you. In your mind, you are going to group them together because they appear to be going in the same direction at the same speed. Phi phenomenon, also called the stroboscopic effect, and we're going to come back to that word a little bit later. The phi phenomenon, imagine this hollow one was a light bulb, and it turns off and on, then this one turns off and on, this one turns off and on, and you're going to see the illusion that this arrow has moving lights. And in fact, I don't think you've ever seen anyone move or run on a phone, computer, or TV screen. Even when you're watching a video, you're not moving anyone. You're not watching or perceiving. You're not, let me rephrase that. You're sensing blinking lights. You're sensing flashing bits of color. You're perceiving it as a whole picture, and you're perceiving an action, someone moving, running, or jumping. Okay, so those are your Gestalt principles. Now, how is perception and attention interwoven? Well, I don't think, without attention, I don't think you can perceive anything. You might be able to sense something, but I think attention is necessary for perception. And the example here is called the figure ground. The face vase illusion. I'm not sure it is, well, we could call it illusion right now. If you're focusing on the figure, which is the vase, you can't perceive the background, the two faces. If you're looking at the two faces, you can't see the vase. And when I teach this, very often I get someone to say, yes, I can, and they're convinced that they can. We're going to learn in a later unit that you can only pay attention to one thing. There's no such thing as multitasking. And so what you think when you're seeing a vase and a face, you think you're seeing them both at the same time. It's very rapid, what's called task switching. And over here, I think the figure is a woman's face. Perhaps the background is a cartoon character playing a saxophone. Can you see that? You can't see both at the same time. So this just shows you that where your attention is, that's what you perceive. Where your attention is, that's what you perceive. Okay, we can't talk about perception without talking about depth perception. And similar to the Gestalt rules of how our mind unconsciously organizes sensation to make sense or perception, our mind uses several cues that our ears and eyes bring to it to make sense to say, where are we in relation to other things, or where are other things in relation to us? We have four quadrants. I've blocked them off so we can pay attention to one at a time. First, let's look at the column. We got monocular. Even though we're going to talk about ears, we're going to call this monocular because most of what we're going to talk about is vision based. But even when we're going to talk about audition or hearing, if you only had one ear, okay, you can still use what we call an external cue or a monaural cue. A monaural, mon means one, oral means hearing. An external cue to determine where something is. So imagine if you only had one ear and you hear something approaching you and then getting even to you and then passing you by like a car. So imagine what's called the Doppler effect, and I'll try to imitate it. If you hear a car go, meow, what you're going to notice is the volume increases as it's getting closer to you. And then what you're going to notice is the pitch increases. So both the volume, amplitude, and the pitch increase as it gets closer to you, and then it decreases as it gets further away from you. And that really only takes one ear to do. Now we're going to move to the idea of, well, let's use the advantage of having two ears. If you have two ears, that means that if someone on your right is talking to you, the vibrating air molecules, their voice is going to hit your right ear a fraction of a second before it hits your left ear, which means your ears are going to send your brain two different signals. But you don't notice an echo when people talk. What your brain does is merge them, but it uses the difference in that signal And that's why you can usually tell where someone is in relation to you with your eye, even with your eyes closed. And if you don't believe me, I'd like you to do a little mental rewind. Go back 10 years when you were in the shallow end of the pool playing Marco Polo. When someone said Marco, you could immediately turn your head and know where they were. Okay. Most of depth perception cues for the AP curriculum are centered on vision. If you only had one eye, you can still have an idea of where something is in the world, even though you don't have both eyes. So let's look at what's called the relative size. And it's real. I'm going to go through these really quickly. I'm not going to overteach them. We have a lot to do. And again, this is a review video. I think many of you have seen this before. So relative size, the smaller something is, the further away it is, and the further away it is, it's going to appear smaller. Height and visual field. Well, if something's far away, it's going to appear higher in your visual field than if it's closer. And if you don't believe me, I urge you to try to draw like a landscape. And you're going to notice you're going to draw the sun, which is obviously far away at the highest end of the highest part of the picture. Interposition, things that are closer are in front of things that are further. Okay, pretty self-explanatory. Linear perspective, if you ever try to draw a cityscape, you're going to try to draw the corners of the buildings to give you an idea of what is front, what is back, or if you draw, the landscape idea of a disappearing road into the horizon. I think linear perspective gives us an idea. Now let's look at number one and number two, relative size and linear perspective. After this, or maybe right now, make a note to search up the Ames room. The Ames room illusion is a carefully constructed room that it looks like somebody gets miraculously bigger and someone gets miraculously smaller because they're going to, the architects of the room play with the idea of linear perspective and fool the eyes. Remember, what you sense isn't what you perceive, and the Ames room shows that very nicely. Atmospheric cues. This means that, well, there's moisture, water vapor in the air. There's moisture in the air. There's smog in the air. So things that are further away are going to look a little bit blurry. If you look at a mountain on a particularly hot and humid summer day and the mountain's a couple miles away, the mountain's going to appear blurry. That's going to give you a clue, even though you only have one eye. Light and shadow. I think you can understand how light and shadow would give you an idea of where something is in the world. Texture gradient, things that are further away look smoother. Things that are closer in look a little bit rougher. Okay. And motion parallax is the idea that if something is moving across your visual field, if it's close to you, then it's going to appear as if it's moving very quickly. If it's far away from you, then it's going to look like it's moving very slowly. And if you've seen a jet flying in the sky near your house, it appears that it's moving slowly, even though it's going several hundred miles an hour. Similarly, if you're in a car and you look out your window, the little lines on the road that are close to your car appear to be whipping by, but that house that might be 200 or 300 feet away appears to be traveling slowly, even though they're both going, or your car is going at constant speed. Okay, binocular vision, obviously you're going to compare what the two signals from your eyes. Remember, you have two eyes, they're each sending a signal to your brain. Your brain takes those signals and compares them. And we call that retinal disparity. Remember, disparity means difference between the retinal signals. So for this, we're going to actually try something. It's the one demonstration I think I can do. So what I'd like you to do is put your hand up to your face right here. And now that your hand up to your face, close one eye, close the other. And then you're going to see two completely different images. Don't believe me? Try it again. Close your right eye, close your left eye. One eye sees the front of the hand, one eye sees the back of the hand. They are completely different and they can't get closer. So what does this mean? The bigger the difference between the two retinal signals, the closer the object is. I'll say it again. The bigger the difference between the two retinal signals, the closer the object is. You don't believe me? Okay, look far across the room now. Stop looking at your screen, look above your screen, and pick a point across the room. Close one eye, close the other, and you're going to see there's virtually no difference. Okay, and that's the idea. The smaller the difference, the further the thing away is. Okay, muscular convergence. Well, we know that our eyes are controlled by muscles, and if you keep your eyes at here and you look up, your muscles pull your eyes up. You look down, your muscles pull your eyes down. Well, if you look at something close in, if I were to bring this pen close in, my muscles are going to push my eyes this way. Just like when you close your eyes and move your arm, you know where your arm is in space. your brain is going to take feedback from the muscles of your eyes and say, wait a minute, if they're flexed a certain way, that must mean the object is close in. Okay, so we went through quite a bit here. If you're a little bit rough or shaky on this, I hate to ask you to come back and rewind or maybe open up your textbook, but there's a lot of dense material in this chart. And it's worth knowing because it tells us again, how does our mind, or I'm sorry, how do our eyes send signals to our brain, and our brain and our mind take those signals and process it into a way that makes sense of the world. Okay, we hit a lot there. Okay, we hit gestalt cues, and there's quite a few of them. In this video, we only hit a few of them, some of them. And then we talked a lot about depth perception cues, and we talked about the idea of how our brain organizes. We looked at the law of Prägnanz, okay, and which is very similar to the idea of gestalt and the binding problem, which basically means we have these isolated, disconnected bits of neural energy hitting our brain. And remember, our eyes don't see anything. Our eyes don't know what to do. Our eyes just say, I don't know, I saw something, I reacted to something, I'm going to send that up the chain and let's hope someone else figures out. Well, your brain gets and say, well, what does that flash of light mean? And that's what the binding problem is. That's the process of transduction, where you take sensory information and turn it into, at first, neural energy. So light becomes an action potential, and then that action potential becomes a thought. Okay, and that's the idea of transduction. Um, I think I just said that. So transduction again, the process of turning environmental energy to neural energy, which is action potentials, right, to perception, to thought. And I don't think you can think a color you've never seen. I don't think you can, convey meaning. I dare you to think of something you can't think of, and all of your thoughts are based on perceptions, and all your perceptions are based on neural energy, and those are of course based on sensations. Okay, let's look at absolute threshold. And that's the lowest level of a stimulus that we can reliably notice. And if I should put in italics, reliably. So I wonder if you've ever had the experience, I have, and maybe this is going to betray how weird I am, that I feel like my phone just vibrated and I check my pocket and my phone's not there. Or maybe I hear a ringing in my ear and I'm like, wait, am I sensing? Am I perceiving? See, our sensory systems aren't perfect. And maybe you felt something that wasn't there, you heard something that wasn't there, you saw something that wasn't there. So if our sensory systems aren't perfectly reliable, in order to define whether or not you heard something, I put that up in quotes or italics, or you saw something, we're going to say that you need to see it reliably. Imagine a third grader getting his or her vision or hearing checked, and they got headphones on just like me. And the technician says, raise your right hand when you hear it. And the technician might even fake and look like they're pressing a button. And a third grader who's easy to please, happy to please an adult is going to raise their hand and say, I heard it. Well, they didn't hear it, or did they? And so what we want to do is make sure that perception is reliable. And that's called absolute threshold. You need to hear something 50% of the time. Gustav Fechner is a psychophysicist from the mid part of the 1800s. Again, this isn't a history lesson, but it's important to know where these ideas came from. So Fechner was the one that came up with the absolute threshold. It's interesting to talk about type 1 error. If you've thought you heard someone calling your name, but they weren't, that's a type 1 error. As we know, that's called false alarm error. Or if you heard a ringing in your ears, did you really hear it, or are you perceiving it or sensing it? You get the idea that this isn't perfectly reliable. Okay, we looked at absolute threshold. Let's look at difference threshold. Can you notice a change in what you were seeing? Okay, what if I somehow or another, through the when I was recording this, or I'm going live on this, I could turn down the intensity of my camera by 1%? Would you notice it get dimmer? Or what if I could turn up my microphone by 1%? Would you notice it getting louder? Well, let's do it this way. If you're lifting 100 pounds and a butterfly landed on the barbell, would you notice it? Probably not. If you're lifting 100 pounds, would you notice another pound, one whole pound? Probably not. Ernst Weber, okay, and remember we pronounce it Wilhelm Wundt, and Ernst Weber And these are the early psychology came from the early 1800s. The psychophysicist and took some of their work and kind of transitioned into psychology. So Weber noticed a constant that he called K. And in lifting weights, it's about 2%. 2% is what you could notice. So if you're lifting 100 pounds and if someone puts an extra 100 pounds in the barbell, you probably won't notice. If you're lifting 100 pounds and someone puts on 2 or 3, you might notice. Okay, this is called Weber's law or Weber's-Fechner law, which is related to the just noticeable difference. Okay, so how is it related? I like to say the JND, or just noticeable difference, is what we get from Weber's law. It's kind of the product. So if you were to ask me, well, what is the just noticeable difference? And I would say, well, it depends on how much you're lifting. If you're lifting 50 pounds, well, 2% of 50 pounds is going to be 1 pound. So the just noticeable difference for 50 pounds is going to be 1 pound. So it really depends on what the original stimulus is. And I don't think for the exam you're going to need to know the constants for brightness or loudness or things like that. Interestingly enough, we are very sensitive to changes in pitch, which is why perhaps humans enjoy music and other animals don't. Or perhaps you can understand sarcasm, or when your mom calls your name in that particular tone, you better move because that tone means something than a different tone. So we're very sensitive to tone. We're not very sensitive to taste. Okay, so we talked a little bit about just noticeable difference. It's important to note that this doesn't necessarily work for extremes of stimulation. So for instance, if you were standing underneath the engine of a running of a jet airplane and it increased 1%, could you notice it? Probably not, because that's the extreme of our ability to perceive. Staring at the sun. Would you notice if the sun had a solar flare if you're staring at it? I don't think you would. It's too bright. Or how about this? If you have a mouthful of salt and someone added a grain of salt or took a grain of salt away, would you notice that difference? I don't think so. So the just noticeable difference is a product of Weber's law. It depends on what the original stimulus level is. So if you are in a dark room and someone turns on a flashlight, well, you could easily notice that. If you're in a brightly lit outdoors and you're on a field and someone turns on a flashlight, would you notice that? Probably not. So it really depends on what the original level of stimulation was. Signal detection theory. Okay, this is a little bit more complex. It's going to add a layer, add a dimension, more than just, well, can you react to the light or not? Can you react to the sound or not? Our brain and our body is getting inundated with sensations at any given time. I don't know what room you're in when you're listening to this, but maybe there's a fan going. Okay, maybe your laptop has a cooling fan, or you're in the kitchen and you hear your refrigerator hum. You ignored that. Okay, so there's a lot of stuff that's hitting our brain. Signal detection theory is it's not as simple as can you just physically detect it. You've got motivation to notice. You got to kind of want it. I mean, I wonder if I'm talking to some teenagers right now, I wonder if this scenario has ever happened. Your parents come home, they see a sink full of dishes or a dirty house, and they notice you're laying on the couch, laying in your bed, and they come in and they say, "Didn't you see it?" And then you have to be very, very careful. I don't suggest you should say, well, mom, define seeing, because seeing is complex. That's probably not a good idea. But I wonder if your average 15 or 16-year-old literally didn't see, depending on what seeing is, the sink full of dirty dishes. Why? Because our brain can only notice really one thing at a time, and that's where we put our attention. Okay, so motivation to notice in addition to ability to notice. That's the signal detection theory. Okay, let's jump down to sensory adaptation. It just means you don't notice things because you've adapted. And we're going to look at in further videos, we're going to explore this not controversial, but I almost always get arguments from my students when I say, you can't multitask and you can only pay attention to one thing, which is really why texting and driving is just a horrible concept to do. So if you can only pay attention to one thing, we talk about figure ground, that means you can't notice anything that you're not paying attention to. So for instance, your socks. I wonder, as soon as I say that, is anyone here wiggling their toes? As soon as you wiggle your toes and you get distracted and I say, okay, quickly, what is the last 4 digits of your social security number? If you're a teenager, you might not know it. Last 4 digits of your phone number, you probably know it, but you forgot all about your socks. When you put your socks on for a few seconds, you notice it. Eventually you stop noticing. That's called sensory adaptation. Sensory habituation is confusing. It sometimes people get these ideas confused. Sensory habituation is you don't react. Okay, you hear it, but you don't have a reflex. So I would use the example of kids or little children get startled by a loud noise. But if an adult works in an industry or is used to a loud noise, they get used to it. They hear it. They just aren't afraid of it. They don't have that reflex of flinching. And maybe you could apply that to your alarm clock. If you hear your alarm clock, perhaps you can sleep through it. Maybe you don't notice it, or maybe after a while, it just doesn't bother you. You hear it, but you just don't feel like you should get out of bed and turn it off. Okay, we went through a lot of pretty deep concepts there. Um, the idea here is intensity of stimulus, our attention, our motivation, and does the intensity change? These four ideas are what makes sensation and perception possible. Okay, I just looked at the time, so I'm going to try to speed up a little bit. Maybe you want to put me on fast forward, but I'm not sure it works that way. Okay, super quick going through the anatomy of the eye. Okay, this idea is in your textbook, I'm sure, and or you can see it online. So I'm going to go through these words super quick. Are you ready? Cornea. I just tell my students, for lack of a better word, it's a windshield. It keeps bugs out of your eye. Your cornea is your clear part in the front of your eye. The iris is a colored sphincter. Yep, you heard that right. It's the muscle that as it closes together, it's going to let less light in, and as it opens up, it lets more light in. Okay, your pupil is the hole. I'm going to basically, if you're writing down notes really quickly, pupil is a light hole. Cornea, windshield, iris, colored sphincter, pupil, light hole. Lens, clear, flexible M&M. It's kind of the shape, it's oblong shape of an M&M, but muscles, you can see muscles right here are going to stretch it out or let it relax and get its more roundish shape. As the shape of the lens change, that's going to pinpoint where. So if light's coming in, if you're looking at this candle and it's going through your clear windshield, clear colored sphincter, which is through your light hole, and it's on the back of your eye, then you're going to see, depending on where it's focused, is going to kind of relate to how far away that object is. And your lens is the one that bends the shape. Okay, upside down, it's important to note every image we see is basically broadcast or portrayed in the back of your eye upside down. And that's fascinating. We're going to talk about this in maybe on Monday, that even though you see two upside down images, your brain takes that information, processes it, and merges it into one coherent right side up image. Fovea. I have CCC, just a quick little mnemonic. Fovea is the center. It has cones, and cones do color. Fovea is the center of what the retina, and I call the retina the movie screen. And that makes sense because when you're sitting and watching a movie, you're not looking at the upper right-hand corner. Your eyes are always staring at the center of the movie, and that's where the projector is projecting. So retina is the movie screen. Fovea is the center of the movie screen. So notice I have retina underlined and rods underlined. Rods are spread out throughout the whole retina. Okay, and that's important. So rods are all over, rods and retina are all over here. Rods allow us to see in dim light and motion, which is why we can't read out of the corner of our eye. You don't believe me? Take a book. You can put it up in front of you. You can read. Keep your eyes forward. Keep your eyes straight ahead. Put a book up here. Because the image won't hit the fovea, which is the center color cones, and that's also where we see fine details such as letters and words, we can't read in the periphery of our eyes. Okay, so cones, again, color, and then blind spot. This is what we're talking about because I want you to be able to explain it. Where the optic nerve comes in, where my cursor is kind of moving around, there aren't any rods and cones. As a result, if this candle image came and was broadcast or projected right on this point, you wouldn't be able to see it. But what's interesting is we have two blind spots every second of every day that we're seeing. You don't notice them now. If you're staring at this screen to your left and to your right, somewhere in your periphery, you can't see because the light is not hitting what's called rods or cones, but your brain is filling in the gap, thereby showing us the difference between sensation and perception. Even though we don't sense something in our blind spots, our brain fills it in and perceives for us. Okay, going quickly. We know that different receptors, rods and cones, respond to different stimuli. And all you need to know is there are three different kind of wavelengths of light that we respond to. Okay, we've got short waves, which are blue, and I say short because there's not a lot of distance between the peaks. Long wavelengths, red, is a lot of difference, and in between there's going to be green. Okay, so what does this mean? Your cone is a specific cell that's going to respond to a specific stimuli. So what your cone is going to do is send a signal to your cone, your photoreceptor, or your rod is going to send a signal to bipolar cells, then to ganglion cells, and then onto the optic nerve. And let's look at a quick diagram. As light, this is the retina, this part right here, as light passes through, it hits your rods and cones, so named, of course, because a cone is cone shaped. And then your rods and cones are going to send a signal in the opposite direction, and they're going to look at your bipolar, hit your bipolar cells. Your bipolar cells are going to send the signal to your ganglion cell, and your ganglion cell is going to send that information onto your optic nerve. So where does it go once it hits your optic nerve? Well, here's a different image. Your optic nerve is going to cross at something called your optic chiasm, and that's where those two nerve clusters or ganglia cross. And remember, you don't have a thalamus. You have two thalami, right? You don't have amygdala. You have two amygdalae. And so the idea here is the amygdala are going to process the incoming information, and then they're going to send it to the back of your brain, your occipital lobe that Mrs. Fenton talked about, in your primary visual cortex. Okay, it's interesting. We need to talk about Hubel and Wiesel. They found a way to detect impulses from individual neurons in the visual cortex. Okay, think this through. For the longest time, the brain still is what we call kind of a black box. If I were to take my computer apart, I don't see the software. I see wires, I see circuit boards. What these gentlemen did was take one step into uncovering the concept of transduction, and they were able to record a single neuron reacting to a single simple stimuli. We're going to call them feature detectors. Feature detectors are neurons that respond to a very simple stimuli, not a complex shape like an apple or even a single letter, because a single letter is a complex shape. What a feature detector is going to respond to is an edge, line, or contrast. And so when these scientists basically monitored the brain of a cat, and when they showed a particular image to a cat, when it hit a certain point in the cat's eye, these scientists could see which neuron was related to, almost related neuron to neuron, from the back of the eye, the fovea, to the back of the brain, the primary visual cortex. And that's a fascinating idea. Okay, that now we got what's happening in the eye all the way to what's happening in the back of the brain. But remember, that was for simple stimuli. Check for understanding, we went through a lot of vocabulary, anatomy of the eye and anatomy of the brain. It's worth cracking open your book, taking a look at that, and maybe writing out some sentences, not just what they do, but how they relate to each other. Okay, two theories of color. Trichromatic theory is basically means there's three cones that respond to light. We talked about short wavelengths are blue, medium wavelengths are green, and long wavelengths are red, which means we have three types of cones in what, not the retina, in the retina, but the specific part of the retina called the fovea. Remember, fovea, CCC, color center cones. The mix of afferent signals sent from the cones in different ratios can produce the perception of any color. It's interesting, you don't see color. You sense, or you don't sense color, you sense radiation, you sense wavelengths of light. You perceive color. Our perception of yellow comes from the stimulation of both red and green cones. When our brain gets red and green signals, our brain says, oh, I know what that is, that's yellow. The advantage of this theory, remember there's two theories, is that it works for the eyes. It's based on how cones work. But remember, there's another theory. The other theory is the opponent processing theory. And this is when certain neural circuits are excited, they cannot process their opposite colors. For example, when blue circuits are active, you cannot simultaneously see yellow. You can see opposites next to each other, but not at the exact same spot. And I think you've been exposed to this: blue, yellow, or opponents, red, green, and black, white. The reason this is important is it helps us explain the concept of after image. And I'm sure you've played with these illusions. If you haven't, maybe search one up, stare at this image for about 30 seconds, keep your eyes focused on it, focused on it, focused on it for 30 seconds, and then transfer your gaze to maybe a white sheet of paper, and you'll see the opposite flag with different colors, the blue, red, and white. Okay. We went through a lot for color vision. It might be a good idea for why wouldn't you just make a quick chart? Okay, compare, contrast, what are the advantages and disadvantages of each theory? What is the main point? And perhaps even who the important people there are. Hermann von Helmholtz and Ewald Hering. It might be an interesting thing for you to compare that to kind of lock it down in your brain. Remember, you practice as you do, or you do as you practice. Okay, common sensory conditions. Well, we're looking at vision and hearing impairments. Okay. The idea here is when you look at a vision or hearing impairments, if you have a problem, we're going to break it down to mechanical concepts. That means there's something wrong with the sense organ, something wrong with the ear. So if someone's deaf and they go to the doctor and oftentimes they say, doctor, I can't hear, the first thing the doctor is going to check is there something in the ear, and it's usually earwax. Okay. That's different than something called neurological. Similarly, if someone has a vision problem, oftentimes I'm going to call it what's called mechanical. It's something structurally wrong with the eye. And here you have two examples. Myopia is when the pinpoint image, remember that candle, is a little bit short. It's falling short of the fovea on the retina. And that's going to mean you're a little bit short-sighted, a little bit nearsighted. Hyperopia is the idea that your lens is not focusing. Your eyeball is actually too short. Your eyeball is a different shape. So the image isn't where it's hitting is not perfectly focused. And that's going to cause people to struggle, perhaps seeing the board or the chalkboard or driving a little bit far away. So those are two neurological, or I'm sorry, sensory conditions, but it's important to note mechanical, neurological. Neurological would be if there's something wrong with the brain, the processing. The sense organs are fine. They're sending the signal to the brain. The brain's just not doing what it needs to do, or the brain's flawed in some way. And an example of that might be prosopagnosia, where someone can't recognize faces. The eyes are sending the signal, the brain just can't recognize faces, or perhaps what we call blind sight. If there's damage to the occipital lobe, somebody can see lights hitting the eye is sending the signal to the optic nerve. It's just the visual processing area in the occipital lobe isn't seeing it. And what's fascinating with someone with that type of damage, while they can't consciously see, if you were to throw something at them, they would flinch, but they wouldn't know why because they couldn't see. And that just shows you there's different levels of processing incoming sensory stimulation. Okay, the last thing we got to talk about is synesthesia. And while it's common, it's abnormal, but yet common enough that it's not so rare. And one of the problems is, how would you diagnose it? Because if someone has seen the world or perceived the world their one way their whole life, they don't know what they're doing is different. And I think you've seen this before. It's perceiving a stimulus in more than one way. And so you're hearing color or tasting sounds, and there's almost an infinite ways that synesthesia can present itself. I put the word problem in quotes. Okay, the problem isn't at the sensory level. The eyes are fine, the ear is fine, the skin is fine, the tongue is fine. It's just that the brain is somehow or another crossing the perceptual pathways. It's not a problem, because most people who have this condition enjoy it. Number one, that's how they've always perceived the world. And number two, it gives them just a different added way. And it's almost like if you were to take that away from them, if you were to cure this, then they'd see the world slightly differently. They'd almost lose something. Okay. Let's jump onto our exam review practice. We're running a little short on time. We're a little bit over, but we're going to talk about Melly in her first department. I'm going to let you read it, but we present a scenario. We have 4 different concepts and vocabulary words that I need that you want to define, apply, and go deeper in. I'm not going to over talk it, but I really want you to take a little bit of time before you start this. Get a piece of paper, get your, get a fresh window open, type this out. But before you do, start a stopwatch and see how quickly you can do a good job on this FRQ. Thanks for watching. We hope you'll join us again. Be sure to complete the practice question. We'll review the answer in the next lesson. Thank you for watching. Bye-bye.